So good afternoon, everybody. My name is E Robayo. Can you hear me?
Loud and clear.
Okay, perfect. So I'm a second-year PhD student in the neuroscience program. My mentors are Dr. Wistrom Noga and Dr. Govind. And today I'm going to present about the tissue microstructural and biochemical characterization of the brain in people with TBI-related chronic pain. So we are part of the Clinical Pain Research Laboratory, and we study chronic pain after neurotrauma, specifically after traumatic brain injury and spinal cord injury. So chronic pain is a frequent consequence of spinal cord and brain injury, and most studies indicate that about 2/3 of patients with either SCI or TBI will experience pain after the injury. Chronic pain is consistently rated as one of the most difficult problems associated with those injuries, and it has a negative effect on people's mental health, quality of life, and rehabilitation, and it is also difficult to treat. So just to describe a little bit about the problem of chronic pain after spinal cord injury and traumatic brain injury. So from a neurobiological perspective, the mechanisms contributing to the development of chronic pain in this population are heterogeneous. Also, the experience of chronic pain is complex and differs from one individual to another in characteristics such as symptoms and severity. There are many patients from whom no adequate structural correlates for their pain can be found, so there is a need to understand the pathophysiological mechanisms that lead to the development of chronic pain at an individual level, so treatment interventions can be tailored to those specific mechanisms. First, we need to understand the events that occur during and after a traumatic brain injury. So annually, approximately 1.7 million people in the United States experience a traumatic brain injury, and this has been linked to cognitive, emotional, motor, and sensory deficits. Traumatic brain injury can be studied in two phases, the primary injury and the secondary injury. The primary injury occurs following the initial mechanical insult to the brain, and it is characterized by symptoms and events such as skull fracture, cerebral contusion, axon stretching, hemorrhage, and synapse loss. Following this stage, we have the secondary injury that is often characterized by events such as energy failure, neuronal cell death, diffuse axonal injury, oxidative stress, immune response, excitotoxicity. And finally, we have events such as axonal reorganization, neuroplasticity, and remyelination. And this secondary injury can last for weeks, months, or even years. So just a brief overview of the neuroanatomy of the somatosensory system. We know that the anterolateral system carries pain, temperature, and crude touch information, and the spinothalamic tract is part of this system. This tract relays some information in the medial dorsal nucleus of the thalamus, the ventral posterior lateral nucleus of the thalamus, and the ventral medial posterior nucleus of the thalamus. And then they project to the primary somatosensory cortex, the cingulate cortex, and the insular cortex. The primary somatosensory cortex is mostly involved in the more discriminative aspects of pain, such as location and intensity. And the cingulate cortex, as well as the insular cortex, are involved in the more affective processing of pain. So it has been found that a lesion localized to the ventral posterior lateral nucleus of the thalamus leads to the thalamic pain syndrome, also known as the post-stroke pain syndrome. An alteration in the thalamic excitability following injury to the spinothalamic tract in a model of a spinal cord injury underlies the alteration in pain perception. And also that traumatic brain injury induced neuronal damage in the somatosensory cortex can promote astrogliosis and persisting neuroinflammation. And neuroinflammation has been associated with some chronic pain states. So neuroimaging research suggests that the pain is processed in a network of somatosensory cortex, such as the S1 and the S2, also insular cortex, limbic cortex, limbic structures such as the anterior cingulate cortex, and also associative structures such as the prefrontal cortex. And they receive multiple inputs from pain pathways, and they form a network. So in this picture, we can see that this network is involved in the perception of pain. We also have a network that is associated with the modulation of pain. So in our laboratory, we consider that the study of chronic pain requires a multidimensional approach. And for that, we investigated people with TBI-related chronic pain in different aspects, such as the sensory state. And for that, we use behavioral measures. The psychological and emotional state, for that we use psychological assessments and tests. The tissue biochemistry and the tissue microstructure by using neuroimaging techniques. And so just to remember that pain is a subjective sensory symptom that is not visible. So it is very difficult to measure. Now I'm going to explain a little bit about the sensory state and how do we characterize this. So this figure shows the pain distribution, and also pain after TBI usually involves the head region. Other common pain locations were reported by our participants, such as the neck and shoulders with the 73%, the back with the 50%, and lower extremities. The classification of chronic pain falls into three broad categories, and it's important to differentiate between them. So we have the nociceptive chronic pain that is caused by tissue disease or damage. We have the neuropathic pain that is caused by the somatosensory system disease or damage. And we have the mixed chronic pain, which is the coexistence of nociceptive and neuropathic pain. During this talk, I'm going to focus on the neuropathic pain that can be characterized by negative symptoms and signs, such as the loss of sensation and increased thresholds for cold or warm temperature. Also by spontaneous sensation or pain, such as pain attacks or burning superficial pain. And also by evoked pain, such as allodynia and hyperalgesia. So as you remember, allodynia is the feeling of pain produced by a non-painful stimulus, and the hyperalgesia is increased pain sensitivity after a mild pain stimulus. So in order to characterize those symptoms and sensory involved with the neuropathic pain, we use a neuropathic pain symptom inventory, which is called NPSI. And this is a self-questionnaire specifically designed to evaluate the different symptoms of neuropathic pain. The final version of this questionnaire includes 10 descriptors and 2 temporal items that allow the discrimination and quantification of 5 distinct relevant dimensions of neuropathic pain syndromes. And those have been shown to be sensitive to treatment, and they can be used to characterize subgroups of neuropathic pain patients. So those 5 dimensions of neuropathic pain are the burning pain, characterized by a burning sensation, the pressing pain, characterized by a squeezing or pressure sensation, the paroxysmal pain, characterized by a stabbing or electric shocks sensation. The evoked pain is when the pain is provoked or is increased by brushing, pressure, or contact with something cold. And then we have the paresthesia and dysesthesia, which is when the pain feels like pins and needles or tingling sensation. So as you can see in this diagram, we quantified those dimensions of neuropathic pain in our participants. And almost 40% of our participants reported having burning pain. More than 80% of our participants reported having pressing pain. Almost 70% reported having paroxysmal pain. And almost 80% reported having evoked pain. And almost 50% reported having paresthesia and dysesthesia. And it needs to be noted that only one subject did not report any of those subscores. So as I mentioned before, those symptoms and signs can be used to classify our participants. So we use a cluster analysis in order to do that. So cluster analysis can be used to classify the subjects based on those sensory profiles. By using a two-step cluster analysis, we obtain 5 different clusters. As you can see in this dendrogram, we have one cluster, then a second cluster, a third cluster, the fourth, and the fifth. And this analysis was performed in SPSS. So those can be called pain phenotypes. And those pain phenotypes are likely to correspond to underlying mechanisms. So in this pain phenotype number 1, we can observe that it is characterized by no burning pain and minimal evoked pain and no paresthesia or dysesthesia. In the pain phenotype number 2, we have similar to pain phenotype number 1, no burning pain. We have moderate present and paroxysmal pain and some evoked pain and dysesthesia and paresthesia, similar to the evoked pain. In the third and fourth cluster, they also have very specific similarities in the symptoms and dimensions of the neuropathic pain. And this pain phenotype number 5, it's characterized by moderate to severe burning pain, severe pressing pain, severe paroxysmal pain, severe evoked pain, and also paresthesia and dysesthesia. So by doing that, we were able to characterize those pain phenotypes that need to be further validated, and also need to compare with other pain phenotypes of other pain populations, such as the spinal cord injury. So after doing that, we wanted to characterize the psychological states by using psychological assessments. So we focused our attention in the learning and memory function, the attention processing speed and cognitive flexibility, fine motor dexterity, emotional function, and post-traumatic stress disorder, as well as post-concussion symptoms. And for that, we used those questionnaires and tests that were previously validated in previous studies performed by psychologists and other pain groups. And as expected, we found that, for example, the post-concussion symptoms and the post-traumatic stress disorder was increased in our participants with TBI-related chronic pain when we compare with other participants with TBI and no pain and with healthy controls. So here we have a significant increase in the post-concussion symptoms calculated by this test, Rivermed, and then in the post-traumatic stress disorder, also significant increase. In the attention and cognitive function, we also found a significant decrease calculated by this test in our participants with TBI-related chronic pain compared to the healthy participants, healthy controls. And in learning and memory, we found that a significant decrease in the learning memory in our participants with TBI-related chronic pain compared to healthy controls and to TBI without pain. And the emotional function was also altered in our participants with TBI-related chronic pain. As you can see in those scores in the Beck's anxiety test, we have a higher anxiety and depression scores in our participants with TBI-related chronic pain. And here we perform a one-way ANOVA with two-case correction for multiple comparisons. We also look at the relationship between anxiety and depression, and we found that anxiety and depression are positively correlated with pain severity. So as you can see here, as the pain severity increased in our participants, the anxiety also increased, similar to the depression. As the pain severity increased, the depression also was shown to increase. So now, after characterizing the sensory profiles and after we found the different clusters, we investigated the psychological states, and now we wanted to look at the tissue biochemistry and tissue microstructure. And for that, we use neuroimaging techniques. So now I'm going to describe how we characterize the tissue biochemistry by using magnetic resonance spectroscopy. So magnetic resonance spectroscopy can quantify some metabolites that are present in our brain. As an example of those metabolites, we have N-acetyl aspartate. We have GLX, which is a combination of glutamate and glutamine. We have creatinine, we have choline, and we have minoxidil, as well as other residual lipids. So we focus our attention in N-acetyl aspartate because it has been considered a neuronal marker. It is synthesized from aspartate that undergoes an acetylation reaction, forming the N-acetyl aspartate molecule that has those hydrogens that give the characteristics of this peak that is detected by the magnetic resonance spectroscopy. So N-acetyl aspartate is primarily located in neurons, and at decreasing its concentration may indicate loss of neurons or neuronal dysfunction. Lower thalamic concentration of N-acetyl aspartate has been found in people with heterogeneous chronic pain, heterogeneous neuropathic pain, and neuropathic pain associated with diabetes, and also in spinal cord injury. So we compare the intensities, the signals obtained from N-acetyl aspartate in our participants with TBI-related chronic pain, and we compare with participants with TBI but no pain, and with whole body controls. So as you can see, we looked into some regions that are important for the perception and modulation of pain, such as the insula. And as you can see, we found a significant decrease in N-acetyl aspartate when compared the participants with TBI and no pain and healthy controls. However, there is a trend in increasing N-acetyl aspartate when we compare people with no pain and pain after a TBI. And this relationship was not significant, but that relationship suggested an hyper-excitability in this region, in the insula, in those participants. Then when we look at the anterior cingulate cortex, that is also important for the affective perception of pain and also modulation of pain. We found a significant decrease in the N-acetyl aspartate in our participants with TBI-related chronic pain as compared with the healthy controls. And that can suggest a dysfunction in this area in the anterior cingulate cortex. Then we look at the mid-cingulate cortex, and we also found a significant decrease in both TBI groups as compared with the control healthy group. And then we look at the post-central gyrus, which is the primary somatosensory cortex, and we only found a significant decrease in our participants with TBI without pain compared with the controls in the right post-central gyrus. We look at the thalamus. We found a significant decrease in people with TBI no pain, but also a trend in decrease in aspartate in people with TBI-related chronic pain. And finally, we look at the hippocampus, because the hippocampus is one of the limbic areas critically involved in anxiety, depression, and learning and memory. And we only found a significant decrease in people with TBI and without pain. The test that was performed here was one-way ANOVA. We took its correction for multiple comparisons. So now we wanted to look also at the individual values. So as you can see here, in people with TBI-related chronic pain, the values found in the anterior cingulate cortex of N-acetyl-aspartate are heterogeneous. However, when we look at correlations with some of the neuropathic pain symptom inventory dimensions, we found that the N-acetyl-aspartate in some regions, such as the frontal, was positively correlated with this present pain. So as you can see here, and also in the anterior cingulum, as I shown in the previous graph, it was positively correlated with present pain. When we look at paresthesia and dysesthesia, it was also positively correlated with aspartate values in the frontal region, in the insula, in the mid-cingulum, in the hippocampus, in the left hippocampus, and in the postcentral gyrus. So those values suggest that there is some associations between the specific neuropathic pain dimensions that suggest some mechanisms and the values of N-acetyl-aspartate that are the neuronal markers. After this, we also look at other biomarker, which is the glutamate and glutamine. And as you know, glutamate is the major excitatory neurotransmitter. And decreased function of the glutamine, can be caused by neuronal dysfunction, specifically excitatory neurons. So then, sorry, I received a question. So then we look at the relationship between N-acetyl-aspartate and glutamine, and we found that they were positively correlated. So let me check out the question. I will, as soon as I finish this section. Okay, so as I mentioned in one of my introductory slides, not only group analysis need to be performed, we also need to look at the specific neuropathic pain phenotypes and also at the individual level. So in this case, I selected a participant from the pain cluster number 5, which shown the severe characteristics in the neuropathic pain dimensions. So in this case, we have the spectrum, the color-coded spectrum of N-acetyl-aspartate, and we can clearly see that there is a difference between the left and right, specifically in the insula. So here is the peak for the N-acetyl-aspartate, and here is the peak for the N-acetyl-aspartate. So there is a decrease in this, in the right insula, this is the left, in the left insula, as compared with the right insula. Let me see if I can see the question. Okay, so the question was in slide 21, both the neuropathic pain, the no pain and pain groups show significant differences compared to the control, but the effect sizes seem small. It is hard for me to understand, do those changes represent biological meaningful changes? How do we know? Yes, you are right. In order to have a significant effect size, we needed approximately 50 participants in each group. And that's why almost at the end of my presentation, I'm gonna show other statistical analysis, such as the network analysis that we're going to conduct in order to really see biologically meaningful changes. And also, as I mentioned, this chronic pain, TBI-related chronic pain, is heterogeneous, so it's really difficult to obtain group differences. So after that, we wanted to look at the tissue microstructure. For that, we also use a neuroimaging technique. And this is an MRI-based neuroimaging technique called diffusion tensor imaging, and later I will describe another technique. So this diffusion tensor imaging is based on diffusion of water molecules. And in this schematic graph, we're just looking at two sites in the brain. This one is representing the gray matter, and this one is representing the white matter. So in the gray matter, we are expecting an heterogeneous tissue characterized by neuronal cell bodies, astrocytes, microglia. And as you can see, the diffusion of water molecules are going to encounter some barriers. And those barriers are going to correlate with actually the integrity in this tissue. And then when we look at some white matter regions, we're going to expect to find the axons. So in this case, the diffusion of water molecules will follow the direction of this of the axons, and we can also find changes in the integrity of those regions based on the diffusion of the water molecules. So in this graph, we can see that the fiber tracks have an arbitrary orientation with respect to the X, Y, and Z axis. And they impose a directionally dependence, which is called anisotropy, on the diffusion of water molecules, as you can see by this effect. So water molecules will mainly diffuse in this direction, and also in a lower degree, they will diffuse in the radial direction, which is the perpendicular to the direction of the fibers. So those values can be represented as an ellipsoid. So in the ellipsoid, we have three characteristic vectors. This one is the principal eigenvector, which corresponds to the direction of the fibers. But also we have other eigenvectors that will be used to characterize the radial diffusivity. So this corresponds to the principal eigenvector. Then we have this, another secondary eigenvectors. So the most common values used from diffusion tensor imaging are the anisotropy, calculated as fractional anisotropy, and the mean diffusivity that calculates the diffusion of water molecules independent to the direction of diffusion. And those values can be calculated in voxels. Typically, a voxel has those dimensions, but we can set the different sizes for the voxels. In our protocol, we calculated those values in the whole brain. And then we use an atlas incorporated into our program in order to calculate in the specific structures that I shown also for the N-acetyl aspartate. So it has been proposed that neuropathology alters the normal diffusion of water molecules. And in this diagram, I just wanted to compare how different events can alter the diffusion of water molecules. So first in A, we have normal myelinated axons that can be characterized by axial diffusion and radial diffusion. Then we have, in this case, axonal myelin injury that can be, so in this case, we can calculate a reduction in axial diffusion and an increase in radial diffusion. We have axonal and myelin injury with cell infiltration, for example, in the case of astrogliosis, and we will expect to have a decrease in the axial and radial kurtosis. We have axon and myelin injury with axonal loss, and we will expect an increase in radial kurtosis. And finally, in this case of axon and myelin injury with axonal loss with cell infiltration, we will expect a decrease in axial diffusion and an increase in radial diffusion. I received a question, I think. Okay, so thank you. Yes, I will answer all the questions at the end. Yes, diffusion tensor imaging also was correlated with some immunostaining analysis. So in this experiment, they look at intact mouse brains and they stain for the myelin basic protein. As you can see here, they use this method called clarity. They use 2-photon imaging and they obtain this clarity image that shows the direction of the white matter fiber track. And by using the DTI method in each voxel, we can see the principal eigenvectors that I shown in my previous slide. And as you can see, those principal eigenvectors in each DTI voxel correspond with the direction of the white matter track. And those vectors can be also used to later reconstruct the white matter tracks in 3 dimensions. And that method is called tractography. So in this study, they look at a 49-year-old patient that suffered TBI, experienced a TBI. And they imaged this patient at 16 months after the initial injury. So they look at the FLAIR scan and they couldn't find any abnormality when they compared those two left and right regions. However, when we look at the color-coded fractional anisotropy map obtained by DTI, they found a reduction in the fractional anisotropy in this region. They superimposed that region in a T2 weighted image and they reconstruct those white matter tracks. As you can see in this image that was obtained by tractography, they saw that those fibers were discontinuous in this region. So as you can see, DTI can detect abnormalities that are not visible in regular CT and MRI scans. So tractography also confirms axonal injury. So in this experiment, they look at the corpus callosum after also, this was a model of TBI, and they found diffuse axonal injury based on the neurofilament staining. And when they reconstructed the fiber tracks with tractography, they also found that the tractography shows this diffuse axonal injury. As you can see, the fibers here, there's a decrease in the number of fibers, and also if we calculate the length of the fibers, we're going to find a difference between the pre-injury and the post-injury. So a Gaussian distribution is assumed by the diffusion tensor imaging. That means a zero kurtosis. However, it has been found that the diffusion of water molecules in biological tissues follows kurtosis distribution, which is a non-Gaussian distribution. So kurtosis is a dimensionless higher order statistic value that increases with the complexity, and that means impediments to the water diffusion. And in biological tissues, that will signify, for example, organelles or membranes. So this graph illustrates the errors associated with the assumption of a Gaussian distribution of water diffusion, as in the case of DTI. So we're looking at measured data at different B-values. So B-values are the diffusion sensitizing values. So if we calculate at B-values of 1000, the measured data can be fitted with both models, the diffusion and the kurtosis model. However, when we increase the values, the B-values, as in this case 2000 and higher than 2000, only the diffusion kurtosis model can represent the measured data. So by this model, we have an improved ability to resolve the intravoxel crossing fibers, resulting in an improvement of the white matter tractography, and that adds an specificity and sensitivity. So in order to have the diffusion kurtosis model, we need to add some elements to the equation that is used to calculate the metrics, such as the axial kurtosis, the radial kurtosis, as well as the fractional anisotropy that I mentioned before, and mean diffusion. So this diffusion kurtosis metrics are able to estimate the complexity of environment. And in this example, I'm showing a normal axonal integrity. And axial kurtosis will be measured in the principal axis of diffusion. And the radial kurtosis will be measured in the perpendicular axis of diffusion, perpendicular to the main axis of diffusion, sorry. So in a case of abnormal axonal integrity, as represented here by demyelination, for example, or axonal injury, we're going to observe lower axial kurtosis and lower radial kurtosis, respectively. And those values imply less impediments to the normal diffusion and a reduced complexity of the environment. And this other case of abnormal axonal integrity that is represented by in this case, scar formation or astrogliosis, glial scar formation or astrogliosis, we're gonna find higher axial kurtosis and higher radial kurtosis, as well as higher mean kurtosis. And those values imply more impediments to the normal diffusion and greater complexity of the environment. So diffusion kurtosis imaging was also used in a preclinical model of TBI. So in this experiment, we have a mild controlled cortical impact injury. So here we have compared with the sham animal. So this is the injury site. And they observed that there is an reactive astrogliosis that is found after the injury in the ipsilateral side and also in the contralateral side. And they use both DTI and DKI metrics. So here we have mean diffusion that was obtained from DTI, and we have mean kurtosis that was obtained by DKI. And they found that the increase in mean kurtosis was associated with the increase in reactive astrogliosis. And that increase in mean kurtosis suggested increase in the complexity of the environment, as you can clearly see in those images. So diffusion kurtosis imaging has been used also in human pain studies. And this is a study of patients with post-neuropathic neuralgia that were compared to healthy controls. They look at the pain scores by using this visual analog scale. That is a scale of 0 to 10, and 0 being no pain and 10 being the worst pain possible. They found that an inverse negatively correlation between the axial kurtosis and the pain scores in the left and in the right insula. And those results suggest that decreased complexity in the insula in the axial direction was correlated with increased pain scores. So we also investigate those values in our participants, and in those two graphs, we compare our participants with TBI and pain, with the participants with TBI and no pain, and with healthy controls, in only a couple of regions. And we found a significant decrease in the cingulum, in the right cingulum, when compared the people with TBI, no pain, and healthy controls. However, there is a trend in the participants with TBI-related chronic pain as compared with the controls. And similar to those results we have in the superior corona radiata, we can see a decrease in both the TBI groups as compared with the healthy controls. We also look at the radial kurtosis, and we found similar results. So this reduction in the kurtosis values suggests less complexity in those regions that can be caused by neuronal injury or demyelination that can occur after the traumatic brain injury. So we also quantified the mean kurtosis in some regions of the gray matter. And here I'm showing the individual values of our participants with TBI-related chronic pain compared to the healthy controls. And you can see that those values are also heterogeneous. We can see some decreases, we can see some increases. And when I performed the one-way ANOVA, I found that it was only significant between the healthy controls and TBI with pain in the right thalamus. So only here there was a significant difference. So I would like to mention again that it is important to also look at the individual the changes at the individual level. And for that, I selected again the same participant, participant number 9, that was part of the pain cluster 5, that presented the most severe neuropathic pain symptoms. And as you remember, the N-acetyl aspartate was found to be increased in the left cingulate. So in this case, I analyzed some of the measures. Those are from the DTI, those are fractional anisotropy and mean diffusion. And it's really difficult to see differences in the regions when we compare the left and the right. We can also do some comparisons with a control subject, but that's part of our future analysis. And when we look at radial and axial kurtosis, as you can see, it's also difficult to find any differences between the left and right. However, we're going to do some future analysis that I will mention in the next slides. So as some of our conclusions, I want to mention that the pain phenotypes can be identified based on the sensory profiles of each individual. And those pain phenotypes are thought to represent the specifically mechanisms that are involved in the development of chronic pain in this population. Also, we found that altered psychological states and also emotional states in people with TBI-related chronic pain compared to people with chronic pain, with TBI no pain and with healthy controls. However, we need to do further analysis in order to characterize those states. And people with TBI-related chronic pain presented biochemical and microstructural abnormalities in the brain areas involved in the modulation and processing of pain. But we're gonna use different approaches that I will mention in my next slide in order to really characterize the biochemistry and the microstructure in those participants. So our future approaches, we're going to use a network analysis by using this multimodal network analysis toolbox that also employs tractography. And with this, we can actually construct a network of regions involved in the perception modulation of pain, and also look at the connections, neuronal connections between them. And that will also generate a model that can be used to test, use other statistical methods. And also tractography, as I mentioned before, is the reconstruction of the fiber tracks, and that can help us to visualize the lesions in those fibers. So I made those drawings using the SCI Studio software, and I use data from the Human Connectome project that is available for us to use. So I just need to mention that the long-term goal of our proposed research is to develop a basis for translational research and treatment interventions tailored to specific pathophysiological mechanisms and psychological contributors of TBI-related chronic pain. So now I would like to thank my mentors, Dr. Wisranoga, Dr. Govind, and all the clinical pain research laboratory members and collaborators, Roberta, which is a postdoc in our laboratory, Lorianne, who is a senior research associate, Suleiman, thank you for providing some of the data for the biochemical and microstructural analysis. Also Madison, Tope, and Tyler, who are the undergraduate students that worked with me. Also, I would like to thank the University of Miami, the Miller School of Medicine, the Miami Project to Cure Paralysis, all the TBI and healthy control participants, the neuroscience graduate program, Dr. Colleen Atkins, Maya, all my neuroscience friends, and the PIPS program, Dr. Salas, Laura, and all my PIPS friends. So now, thank you very much for your attention, and I can take any questions. I have a question for slide number 24. Sorry, let me go to slide number 24. Slide 24. Okay. Oh, okay. So this question is regarding the connections between the points, different brain regions. Okay, yeah, so I added those connections between the points to actually see for each of the participants. So for example, I wanted to compare the right and the left side. So in this case, this is the same participant and it has like a decrease in both regions, the right anterior cingulate cortex and the left anterior cingulate cortex. So, but if we look at another participant, we can see that only the left side is affected. And also in the post-central gyros, similarly in that participant, only the left side is affected. And because we don't have the information of the site of injury, I was interested in looking those differences between the left and the right side, and also between the different regions. So in this case, like an decrease in the cingulate cortex, but that participants have like normal, if you can consider normal glutamate values in the insula. I hope that I answer your question.